Hopefully you had a good little break. I'm Shannon Wattis. I'm the Associate Director for Rivers Edge West, and we are on our last session of the day. It should be fun. So this is the poster session and student contest. This is kind of our best efforts at recreating our live poster session, so we hope you enjoy it. I'll give you a little rundown on how this is going to work, and then we'll go right into our poster presentations. We don't have a lot of time here, so this is going to run pretty quickly. So we have 5 poster presenters, 3 of which are students. Each presenter will have 3 to 5 minutes to present, and then we'll go from one presenter to the next with no questions in between. So, and if you're interested in looking at the actual posters, you can look at our conference agenda, and the poster links are in there as well. Let's see, then after that we will vote on your favorite student poster. So I'll make sure to announce which ones are students as we go through these presentations, but make sure you pay attention to the students. We will then go into, we'll do a poll, so we'll announce the winners. That'll be fun. So first place is going to get $150. Second place is going to get $100, and then third place is going to get $50. So looking forward to that. And then we will actually go into some breakout rooms. So we'll give you guys an option to pick. You can kind of jump around to different breakout rooms depending on which poster presenter you want to ask questions of. So I'm going to go into how this is going to work. So once we're done with all the presenters and we voted on the students, you're going to get a prompt that says join a breakout room. So it'll be down at the bottom of your screen. You can click on the breakout room icon, and then it should pop up with the list of presenters, and you should be able to click on which one you want to enter, and it should take you to that breakout room. If you don't see this little join breakout room button, you should still be able to click on the breakout room icon and still get in there. If you're still having issues, just put in the chat box where you want to go, and we'll get you in the right place. Okay, so once you are in a breakout room, if you want to switch and go to a different breakout room, you're going to click on the Leave Breakout Room button. Do not click on the Leave Meeting button. That will kick you out of the conference. If you do that, you can just click right back in, no big deal. And then once you leave the breakout room, it's going to take you back to the main screen, and then you can choose a different breakout room. We only have about 10 minutes on the agenda for the breakout rooms, but we're going to allow them to run as long as you guys want. So feel free to hang out, talk. We won't close them right at 4, so just so you know. I did want to make an announcement that Dave and Peyton both have limited time, so if you want to ask questions of either of them, join their breakout room first before joining any of the others. Okay, so with that, the first presenter that we have up is Dave Bastian, and he is going to talk about the One Riverfront Commission. Dave, you can go ahead and share your screen.
All right. Let's see. We rehearsed this, so it should work flawless, right? So I am Dave Bastian, and I am not a student, so you don't have to pay attention to my presentation like Shannon said. I'm a One Riverfront Commission member, and I've actually only been on the commission a short time. Moved here recently, just a few years, and I was only on the commission for a few months before COVID kind of, curtailed a lot of our in-person gatherings. So I'm a tiny bit nervous about talking about this because I don't consider myself an expert on this or really any subject. But for our story today, we're going to start in the 80s. Grand Junction had a nickname at that time, and it was a nickname that we're not fond of. Let's put it that way. But unfortunately, if you were coming into the area from a lot of places, especially you can see here in this photo, it was a nickname that was sometimes apt. We had junk essentially all over the river, jamming up the river. Riprap, cars that were forming as riprap and a tailings pile in one case. In addition to all that, which wasn't really encouraging use of the river, there wasn't much in the way of access to the river. There really just was not a way for the public to access the river at all. Now, at this point, efforts were underway to kind of reclaim some of this space, and parks had been created here and there, but really more needed to be done. So in 1987, the One Riverfront founders, they led this effort to clean up this place called Watson's Island, removing the junked cars, dealing with the uranium mill tailings. If you were to visit this place today, especially if you're like me and you haven't lived here a great deal of time, it's hard to even imagine this could have looked like a junkyard, essentially. I mean, now it is trails, it is a disc golf course, there's art installations here and there. In fact, I walked my dog there this morning, and my daughter and I often go biking at a beautiful little bike park really close by. So a lot has changed. So this incredibly successful endeavor on Watson's Island really is what led to the creation of One Riverfront. That same year, 1987, the commission was formed by the city of Grand Junction and Mesa County with the direction to celebrate and preserve the cottonwood groves, wetlands, cattail marshes, gravel bars, and islands of the Colorado and Gunnison rivers. And at that point, the Colorado was called the Grand. Well, not exactly at that point, but it was at one point. So the One Riverfront is an all-volunteer commission and foundation. We have one staff person who oversees the administrative side of things. We meet our mission by developing the Riverfront Project, and that's something that's often endearingly described as a string of pearls. Those string of pearls are county properties, city parks, state parks and wildlife areas, and open space that are connected by multi-use trails that stretch along the Colorado River. And we also celebrate this by holding a summer concert series and really getting involved in a lot of local festivals and events that happen and sort of to tell our story. So in 2014, big milestone occurred. That was when the trail between Grand Junction and Fruita was completed. This was a milestone not just for One Riverfront, but really for the entire valley. It's also, I think we like to describe it as an opportunity to really showcase what can be done through collaboration and partnership and the cooperation of local and state government, private property owners, the nonprofit organizations, including River's Edge West that support this endeavor, service clubs, community volunteers, foundations, and financial supporters. So this whole endeavor was really born out of a community need, and those of us who are on One Riverfront, we serve that need. It's important to note, I think, that this is not a publicly funded trail. This is a community-supported trail and a community-supported effort done, essentially by the partners of One Riverfront. I have, in my life, experienced the fact that trails make life better. A day that begins walking along a trail along a river is just going to be a better day. And in my time here in Grand Junction, I've really watched this community improve. Like I said, I've only been here 2 years, and I've seen some incredible improvements. This trail here is a perfect example of that, going from downtown to the very popular Lunch Loops biking trail. So you can get on a bike and ride all the way there, ride while you're there, ride home. Trails make life better. And One Riverfront has really been at the forefront of helping to make those things happen, make that better. I love, anyone who's ever heard me talk has probably heard me give this quote because I love it. It's a Wallace Stegner quote, and I think it really sums up One Riverfront and in really any of these endeavors. One cannot be pessimistic about the West. This is the native home of hope. When it fully learns that cooperation, not rugged individualism, when it fully learns that cooperation, not rugged individualism, is the quality that most characterizes and preserves it, then it will have achieved itself and outlived its origins. Then it has a chance to create a society to match the scenery. And that's my favorite part, I think, just that goal of trying to create a society to match the scenery. So that's it for me. Thanks.
Good job, Dave. Even though you aren't a student, it was really
I'm not, you can tell I'm not a student with.
All right, very good. Up next, we have Margot Mason. She's a student with the University of California, and her poster title is Soil Analysis for Restoration on the Santa Clara River.
Okay. Sharing my screen. Can everyone see that? Okay. So, yeah, okay, 1 second. Okay. Oh, there went full screen. Okay. Hi, guys. So the Chesapeake Cienega is the site of an important groundwater-dependent ecosystem on the Santa Clara River, which, despite being one of the most hydrologically intact and biodiverse riparian corridors in Southern California, is still threatened by agricultural impacts and rapid urbanization. Understanding the abiotic conditions is important to making informed decisions for any restoration project, and is especially crucial on the Cienega, where flooding and past uses of watercress farm have created diverse soil conditions. So, to assess the soil conditions on the site, we collected and tested 60 soil samples and analyzed the results in 2 ways. First in R to get a sense of any trends on the site, and second in ArcGIS Pro to identify locations where soil conditions were appropriate for different native species. The sample sites were chosen based on a 150-meter grid, but were adjusted to include areas with different physical characteristics, such as the old watercress beds and the berms in between. At each site, 3 near-surface samples were taken and tested for pH, salinity, soil texture, and bulk density. In R, these variables were tested against 3 potential predictor variables: land use type, distance from the river channel, and scour history. It was found that distance from the channel was significantly related to higher pH, shown in orange, and higher salinity in blue here. Although further analysis showed that clay content also increased with distance from the river channel and was a confounding variable in these relationships, distance could nevertheless be used as a proxy at the Cienega for these 2 soil characteristics. As a separate approach to analysis than the modeling in R, we also mapped the soil sampling results in ArcGIS. First, maps for each variable were created using inverse distance weighted interpolation, like this one for pH you can see here in figure 2. Because of the abrupt transitions on the site, the area of the old watercress beds here was interpolated separately from the rest of the site. So that's the reason for some of the abrupt transitions you see. These maps could be used to guide planting on their own, but to take it a step further, I used spatial modeling to identify areas with the necessary soil conditions for desired native species. To do this, first, decent polygons were generated around each of the soil sampling locations and were altered to follow the geographical barriers of the site. So, for example, the polygon around this soil sample in a watercress bed only extends in the watercress bed. The soil conditions in these polygons were combined with this model of water availability on the site, which was created from shallow well data and elevation data. And together, these created maps of locations appropriate for different native species. So, for example, this is the map for California sagebrush, which requires a pH of less than 8, low to moderate salinity, and relatively dry conditions. So the ideal locations are shown in green, and adequate ones are in orange. If you're interested in this approach to soil analysis or how the water availability map was created, this QR code here links to a document with a much more detailed walkthrough of the process I use, and you can also ask me any questions in the breakout room.
Great job. Okay, we're going to keep this this party moving here. Next, we have David Moore and Tori Barron. They're with the Bureau of Reclamation Technical Service Center. Their poster title is Results of 17 Years of Monitoring at the Las Lunas Habitat Restoration Site in New Mexico.
Okay, thanks, Shannon. Yep, as she said, my name is Dave Moore. I'm with the Bureau of Reclamation. I'm not a student, but if there were a prize to win, it might be for the busiest poster ever. Can you all see that screen? What are you looking at here? It doesn't seem like it's sharing right.
We can see it.
Can you?
Yeah.
Okay. Okay, so you see my mouse moving around here?
Yep.
Perfect. All right. So I'm going to zoom in. Basically, the Los Lunas Habitat Restoration Project was installed along the Rio Grande in response to, a general lack of riparian habitat in the entire southwestern United States. It was an area, and the work was done primarily by the Corps of Engineers and the Bureau of Reclamation. It was a kind of a collaborative project. So it was an area that had burned, and somebody got the idea, hey, let's go knock all those snags down and do some work and try to create some habitat for a couple different species, one being the southwestern willow flycatcher and the other being the Rio Grande silvery minnow. The yellow-billed cuckoo wasn't listed yet, but ultimately it was factored into some of the work that we ended up doing there. And then a year or two later, we began monitoring the area to see if we were being successful at kind of the goals of creating riparian habitat. There were two different treatments within the Los Lunas restoration site, and I can show you this picture here. I don't know if you can see very well, but it's divided roughly down the middle of the site, kind of longitudinally, to where on the western side of the site, adjacent to the road, the burn was left as is. And then on the eastern side of the site, the area was cleared and there were some plantings done. But mostly it was just kind of natural regeneration was allowed to take place. So we've got two different portions, the burned area and the cleared overbank area. So in order to monitor the progression of the site, we determined we should look at birds, we should look at veg, and we should look at groundwater. So our avian monitoring consisted of point counts that were, pretty standard 5-minute, 50-meter fixed radius point counts. And then we also did flycatcher and cuckoo surveys within the site to determine if occupancy had occurred at any point. In order to monitor the veg, we conducted a couple different kind of methodologies. We installed some vegetation transects that were monitored on an annual basis, and we also installed some quantification plots that were installed in areas that we thought looked the most suitable for willow flycatchers. And obviously, initially, they didn't look like anything because half the site was bare ground. Then we also installed a series of groundwater wells, within the site, along 3 different transects, basically, that kind of bisected the site from the road to the river. So there was a series of wells that theoretically would be more or less influenced by river flows, and those were monitored with a data logger. So basically, by means of results, and like I said, this if you all want to talk about this, hit me in the breakup room because there's a lot of information here. The site did pretty well in terms of avian monitoring, and I guess I should go back and say we monitored this site for 17 years. It was installed in 2002, and the last year we monitored it was 2019. So got a boatload of data. Our point count monitoring showed that a pretty diverse bird population arrived in the site once the habitat grew up. And basically, over time, the cleared overbank area began to approximate the burned area, which means ultimately, things started looking pretty similar between the two, which is kind of what we were hoping for. The mid-story guild of birds arrived within that overbank area. I think I'm getting messages here. And then, unfortunately, we hadn't found any flycatcher or cuckoo occupancy within the site, but things are still developing, even though we're not monitoring it anymore. During the vegetation monitoring, as you can see here, cottonwoods and willows were dominant, and over time, the total cover increased to where, the last year of monitoring, it was upwards of 90%. So things are, again, starting to look pretty good within this site. Let's see. Groundwater monitoring, not surprisingly, you know, the higher the water table was within the site, the higher amount of overstory cover we recorded. Water table levels were obviously very linked to river flows. You know, more water in the river, the shallower the water table was. And lastly, one methodology that is pretty neat, but doesn't give you a whole lot of, you know, quantifiable data, was photo stations we installed. And this was 2003, right after the cleared area had been cleared. Here you see 2008, 2014, 2019. And things progressed and are native dominated. They're looking awfully good at this point. Unfortunately, there's no more funding or any more mandate to monitor the site. But hopefully one of these days we'll get back in there and have a look at it, because it's looking awfully good. Let me see.
Thank you, David.
Yep.
Great presentation.
Cut me off.
I'm cutting you off.
Like I said, tons of information here, not geared towards this format at all. But if you have any questions for me, just hit me up in a breakout room.
Yep, absolutely.
Okay, yeah, you stopped me. Good.
Okay, up next we have Alma Anitez Morales. She's a student with the University of Arizona. Her poster title is Developing a Land Suitability Analysis for Green Infrastructure Placement in Ambos Nogales.
Okay, let me share my screen. And are you seeing my screen?
Yes, looks good.
Okay. Well, good afternoon. My name is Alma Anitez Morales, and today I will be presenting on a binational research project that I am a part of and taking place in Ambos Nogales. So Ambos is a term used to collectively refer to both Nogales, Sonora and Nogales, Arizona. These two cities sit in the northward gradient Nogales watershed. And during periods of really heavy rain, typically occurring during summer monsoons, surface runoff from arroyos form. And in steep areas located in the west and southern tips of the watershed, these runoff concentrate, reaching dangerous volumes and velocities. Sometimes the runoff combined with untreated wastewater, resulting in a public health risk due to the high pathogen concentrations. They go through the urban areas, which are impervious surfaces. And in this picture right here, you can see a picture from a flood back in 2008, where the intense flooding got into some of the local businesses, resulting in economic loss. And unfortunately, these intense floodings have also resulted in human loss. Green infrastructure solutions have been proposed as way to control this stormwater. For example, in arid environments, vegetated bioswales use native vegetation to slow down some of the runoff and also aid in pollutant removal through vegetation uptake. So our objective of our project is to find which areas in Ambos Nogales will maximize the benefits of green infrastructure solutions. And to do this, we are conducting a land suitability analysis. A land suitability analysis consists of 3 different categories, which are flood mitigation, watershed protection, and green space opportunities. Factors within each category are chosen based on their expected benefits, which I detailed here in dark green, and also on their data availability. Because we are doing a binational project, we are relying on years of binational collaboration and research, which have produced binational soil maps and land use and land cover maps. We're also using qualitative data from interviews conducted with local stakeholders that inform areas that have experienced flooding in the past. We're also using other geospatial data like location of wells, streets, etc. And we're also using outputs from the kinematic runoff and erosion model, KINEROS 2, which is a physically based model developed by the USDA. And it gives us 5 different outputs, which are infiltration, runoff, sediment yield, peak flow, and sediment discharge. Some of the parameters that we used in this model are digital elevation models, which we have gone from different sources, and then we just stitched them together. the land cover and soil maps that I mentioned before, and precipitation data from rain gauges. So we're still working on this land suitability analysis. It's a work in progress, but so far, most of the factors that we have are for green space opportunities. We are still working on the factors for flood mitigation and watershed protection. Here in this map, we show areas that we have identified as ideal for green infrastructure placement so far, but once we have all the other factors, we will revise it. And these areas are in the hatched polygons. So some of our future work include using this final map that we get with and conducting local workshops with residents in both cities so that they can verify whether they think these places work or not. In our binational team of researchers, we also have landscape architecture specialists who are choosing the type of structures, green infrastructures, to be placed. And these structures will be modeled again with Kineros 2 so that we can verify their expected benefits. And with all that, we're going to have the final recommendations for the Green Infrastructure Binational Network. Thank you.
Thank you, Alma. Okay, last but not least, we have Peyton Wills. She is a student at the Colorado Mesa University. Her poster title is Expanding Our Knowledge of the Mitochondrial Genomes of the Tamarisk Leaf Beetles Diarabda species.
Thank you. All right, my name is Peyton Wills. I do research under Dr. Zainab Absoy at Colorado Mesa University. Can you guys hear me okay? I have headphones in.
Yes, we can hear you good.
Okay, perfect. So as Shannon said, my title is Expanding Our Knowledge on the Mitochondrial Genomes in the Tamarisk Leaf Beetles Diarabda species. So tamarisk is an introduced invasive plant species that was brought over to the Northern American continent in the 1800s. It is invasive and damages our riverbeds. And in 2001, the Diarabda beetle was released as a biological control agent. There are 4 known and described species of this Diarabda beetle. And Diarabda, they come from all over the world. So as you can see in this figure on my poster, we have Diarabda sublineata that comes from Tunisia. We have Diarabda elongata that comes from Crete. We have Diarabda carinata, which comes from Uzbekistan, and we have Diarabda carinulata that comes from northwest China area. And the mitochondrial genomes of the species Diarabda carinata and carinulata have already been previously assembled using sequences from shotgun sequencing, which does the entire genome of those 2 species. So the mitochondrial genome was basically a bonus when doing a shotgun sequencing reaction. So the reason why I'm looking and why I'm attempting to assemble and align the mitochondrial genomes of the other 2 species are because the mitochondrial genome has its own DNA. It's different from what makes us who we are, and it's also maternally inherited, and it's in a circular chromosome. So as you can see, so because it is in a circular chromosome and because it's smaller, it has the ability to mutate 10 times faster. And the 4 known species that we have here in Northern America are beginning to hybridize and interbreed with each other. So because that mitochondrial genome is maternally inherited, our overall goal is to figure out and find which set of hybrid mitochondrial DNA is better for evolutionary standpoints. And as you can see in this figure, it's mitochondrial DNA, like I said, is in a circle, and I'm going to represent all of my data in a linear form. So if you imagine me taking scissors and snipping between 1 and 16,000 base pairs and then lining it out, you'll see the diagram right next to it that shows all of the regions of the mitochondria that have protein encoding proteins, tRNA encoding proteins, and rRNA encoding proteins. So this is just a representation for this protein encoding region. It's going to be right there on that same linear map. and that's just helpful to explain my data later on. And so basically the results, what I did is I isolated DNA from the 2 species of Dirabda that have not been sequenced yet, and I isolated the DNA. I then amplified it, but in order to amplify it, you have to have primers that amplify the certain regions of the DNA that you're looking for. So I used a computer software system to design primers, and in figure 5, you can see all of the primers that I designed that worked in sequencing the DNA of these 2 species. So I had successful results of 91% in the Dirabda sublineata and then 84% in the Dirabda elongata species of mapping their whole, of aligning and assembling their whole mitochondrial genome. And the future goals in research of this is to ultimately fill in the gaps of the linear, well, of the mitochondrial genomes, fill in those gaps that are missing, and then annotate them like what is shown in this diagram right here, annotate them with the genes and the protein encoding genes, and then do a comparative study between the 4 different types of mitochondria in the 4 different species. So thank you.
Thanks, Peyton.